Hi, my name is Jake Isanek, and I've been working on a project that applies artificial intelligence to pharmaceutical drug development. And specifically, I'm looking at drug discovery, which is the first step of the drug development pipeline. Drug discovery is where we go from all the molecules out there and we try to pick just a few hundred or a few thousand that we could actually put through to clinical trials. And so this first step determines how long it'll take and how much it'll cost to actually develop a new drug. It shapes just how efficient the rest of the pharmaceutical pipeline will be. And current methods aren't that efficient. It costs anywhere between $160 million and $2.6 billion just to develop a single new drug. That's anywhere from 10 to 15 years of research, development, and testing. And there's two main reasons why current methods are so inefficient. Current methods aren't scalable and they're not multi-objective. Right now, we're relying on basically trial and error to sift through these huge databases of molecules, and that's just not possible. It's estimated there are 10 to the 60th possible synthetically accessible and drug-like molecules out there, meaning trial and error methods just won't ever be practical. They're unfeasible. Current methods aren't scalable. The other issue is that we can't account for multiple properties all at once. When we're developing a new drug, we need to take into account all these different things like absorption, toxicity, and activity on a target, and current methods can't do this. Current methods aren't scalable and they're not multi-objective. And so these two issues are what I'm addressing in my project. To do this, I use a newer approach called de novo drug design, which instead of doing the kind of traditional trial and error, it aims to develop new drugs from scratch. So it creates and generates new molecules and then optimizes them over time. And to do this, I used an artificial intelligence approach called recurrent neural networks, which are able to generate brand new molecules entirely from scratch. I trained my recurrent neural network model on over 500,000 molecules, and it got really good at generating brand new ones. It reached a valid molecule accuracy of over 75%. And so I had this recurrent neural network model generate 20,000 brand new molecules never seen before entirely from scratch. And then I selected the best half of those, the best 10,000 of the 20,000 molecules. And I did this with a non-dominated sorting algorithm that's never been used before. This non-dominated sorting algorithm uniquely allows this algorithm to account for all these different properties all at once and then optimize them. So as a proof of concept, I tested this approach, this non-dominated sorting algorithm, on the rule of three, which is a commonly used guideline in early stage drug discovery. Rule of three has 5 different properties that I accounted for with this algorithm to select the best half of the molecules generated by the recurrent neural network. And then I took this best half and then I retrained the recurrent neural network model on them again. This retraining is called transfer learning and allows us to optimize these molecules over time. And so this creates this cycle where I have the recurrent neural network model generate a bunch of molecules, it selects the best half, and then it retrains on them, getting better and better over time. I had to go through this cycle for 5 different iterations, and I saw huge improvements in the quality of molecules it was generating. The structural diversity of the molecules increased significantly, meaning it found newer areas of the chemical space, and most importantly, the quality of molecules improved 14-fold across all 5 metrics measured all together simultaneously, and that's what makes this approach so powerful in early stage drug development.